50,000 years ago, this wasn't a forest. It was open grassland where ancient species like mammoth, giant elk, woolly rhinoceros, and even saber-toothed tigers roamed. And living among them and off them were our early ancestors, both Homo sapiens and Neanderthals.
We do what we call biomolecular archaeology, which means that we focus on taking samples for DNA analysis.
From the artifacts you find here?
Yes, bone is mostly made of something that is called hydroxyapatite, and it has this specific property that it binds to DNA. It basically sucks DNA and it stores it. With the team at the Max Planck Institute in Leipzig, we were fortunate enough to put together this new protocol that allowed us for the first time to extract human DNA from an object.
Tell me a little bit more about that artifact.
One of the artifacts on which we extracted the human DNA is a wapiti tooth. Wapiti is a kind of deer, it's actually a very big deer. That wapiti tooth was pierced and probably worn as a pendant. The sweat of the person who was wearing it got inside the pendant, and we were able to extract this DNA, put it in solution, and then sequence it.
The ancient tooth artifact wasn't found here, but in Siberia, in what is known as the Denisova Cave. It was sent by its discoverers to the Max Planck Institute in Leipzig for DNA analysis.
When the pendant arrived, it was still covered completely in sediment as it was found. We first want to wash off the sediment and also analyze the sediment. And we want to do this because we want to compare then the DNA that we find in the sediment to the DNA we find in the pendant. And that allows us to make some comparisons that are really crucial to be sure that the DNA that we find on the pendant is really from the maker or the wearer of the pendant.
So that's not just water, that's got the reagents in it.
Exactly. In this case, we are using a sodium phosphate buffer that is able to release the DNA from the pendant without destroying the structure of the pendant.
Once Elena has extracted the DNA, it's time for her academic supervisor, Dr. Matthias Meyer, to analyze it.
What this computer program does, it basically compares every DNA fragment that we sequenced to a database of reference genomes. Each of these lines here, they were identified as looking like human because of that human reference genome here that was used for comparison.
That's our modern-day human up above.
Exactly.
Right. Why is it so important to know about the DNA of ancient individuals?
If you go into time periods before written records of history exist, right, there's just very little can do to understand the past. DNA sort of gives us a way of doing this. So tools for working leather, were they predominantly used by men or women? Hunting tools, were they predominantly used by men or women, right? If you could start to address this by means of genetics, you would start to slowly understand something about labor division in these societies, and that in turn would tell you something about the structure of the society as a whole.